Scientists converted human fat cells into stem cells. First, fat cells are collected from the body and converted into adult stem cells using growth factors. These adult stem cells are then induced into pluripotent stem cells, or iPSCs, capable of developing into any cell type within the body. This breakthrough means stem cells derived from fat could be used to repair various tissues, including injured hearts, damaged spinal cords, degenerated joints and muscles, and even aid in breast reconstruction post-surgery. Their anti-inflammatory properties provide further healing benefits. Because these cells originate from the patient's own body, there's no risk of immune rejection. Fat-derived stem cells can even prevent premature cell death, form functioning blood vessels, suppress autoimmune disorders, and differentiate into nerve cells, offering potential treatments for neurodegenerative diseases like Alzheimer's and Parkinson's. Stem cells can be delivered via bloodstream infusion or localized tissue injections, where they respond to inflammation signals and differentiate into specialized cells that integrate with target tissues or organs. The abundance, accessibility, and lack of ethical concerns surrounding fat-derived stem cells make them a promising source for regenerative therapies. Fats contains up to 90% more viable cells than that of bone marrow, and this concentration of viable cells in fat tissue remains unaffected by age. Fat cells can be turned into stem cells twice as quickly compared to skin cells, and the techniques used for fat cells extraction are easier and more efficient. The process involves a liposuction to obtain the patient's own fat, usually from the abdominal or flank area. The procedure is performed without general anesthesia and is relatively painless. Stem cells retrieved from fats are more easily cultured and grow more rapidly than those harvested from bone marrow. Revolutionary advancements in cellular reprogramming now allow scientists to create stem cells entirely in the laboratory. In a landmark 2006 study, researchers successfully transformed ordinary mouse skin cells into stem cells by introducing just 4 genes. These lab-grown stem cells, called induced pluripotent stem cells, or iPSCs, are as versatile as embryonic stem cells, but created without the need for embryos, bypassing ethical concerns. iPSCs have extraordinary potential. They can be coaxed into becoming almost any cell type in the body. What's more, iPSCs can be generated from a patient's own cells, meaning perfect genetic compatibility and no risk of immune system rejection during transplantation. This paves the way for personalized medicine, from correcting patient-specific genetic errors to testing drugs on cells carrying a disease. Researchers are even building iPSC libraries for disorders like Parkinson's and Alzheimer's. By creating neurons directly from a patient's cells, scientists can study the disease in a dish, test drugs, and unlock new treatment strategies. The rise of iPSC technology has not only removed ethical barriers that once hindered stem cell research, but has also fast-tracked the development of potentially life-changing therapies for a wide range of diseases and injuries.